Hello, everyone, and welcome to today's Drug Hunter Flash Talks. I'm your host, Dennis Koester, and we're happy you're here today. We will be discussing approaches to the treatment of Alzheimer's disease with Don Weaver today. Welcome to the show, Don. We're really happy to have you.
I'm thrilled to be here. Thank you.
Awesome. Without further ado, I want to introduce Professor Donald Weaver. So Don is a very special scientist. He's a senior scientist and the past director of the Krembil Research Institute and has over 35 years of experience in medicinal chemistry and 40 years experience in clinical practice. Don is currently a professor of chemistry, pharmaceutical sciences, and medicine at the University of Toronto, and currently holds the Krembil Research Chair in Drug Discovery for Alzheimer's disease at the University of Toronto. Don obtained his MD in 1981 from Queen's University and a PhD in organic chemistry from 1986. So he's probably the only individual in the world who holds these two degrees at the same time. He was on faculty at Queen's University from 1988 to 2001, where he was the chair of clinical neurology, professor of chemistry and neurology. And then he went to Dalhousie University from 2001 to 2013, where he was the director of research for the Department of Medicine and professor of chemistry and neurology before he moved to Toronto in 2013. His research is focused on the design and synthesis of drugs for the treatment of chronic neurological disorders with a focus on Alzheimer's disease and related dementias. And he uses large-scale in silico computer simulations to explore the structural biology of putative receptors that are relevant to Alzheimer's disease. And he is very passionate about reconceptualizing Alzheimer's disease as being primarily a disease of the brain's immune system. And he will talk about this more today. Don is very, very impressive. Not only does he hold many academic positions, but he also co-founded 6 startup biotech companies, including Neurochem Inc. and Triventis Corp. And he has had 2 compounds reach phase 3 human clinical trials, one of which is tremiprosate, and that is an anti-amyloid glycosaminoglycan mimic targeting protein misfolding and oligomerization. Don has held the Canada Research Chair Tier 1 in protein misfolding diseases and is a Harrington Scholar Innovator. He has also published more than 300 peer-reviewed publications and holds 47 issued patents in drug design and discovery. Don has won multiple awards, international and national, for his research. Don, this is such an impressive CV that you have, and we're very, very excited to have you here with us today.
Well, thank you. And I note in the chat that there are people who were born in 1981. So
wow, that's awesome.
So yes, I will bring my geriatric experience to the conversation today.
Awesome. Yeah. Why don't we get started with some icebreaker questions, right?
All right.
Yeah. What inspired you to start a career, as a doctor and chemist, so in drug discovery?
Yeah. I mean, as you mentioned in the introduction, I first qualified as a physician. So I became a physician first, indeed, a neurologist first. And one thing I rapidly learned that neurology is referred to as the diagnose and adios specialty. We see you, we tell you this is what you've got, but then we go, no, sorry, we don't have any drugs that work. And I wanted to change that. And so, and who makes drugs? Well, it's chemists who invent drugs. And so I went back and did a PhD in chemistry after I had qualified as physician. Not the most financially intelligent move of my life, but from an intellectual and rewarding point of view, it was absolutely the right decision. And as you know, I recently had an editorial in ChemMed Chem in which I discussed this, but more particularly, I discussed why Alzheimer's disease. And early in my career as a physician, I came across an individual whose life had been shattered, and he was reliving it through Alzheimer's disease. And it became apparent to me that this was a disease that I needed to address. And so I've certainly spent the last decades, 3 decades, working on Alzheimer's disease, trying to see if I could come up with something.
Wow. Yeah. No. And we all read your editorial here at Drug Hunter, and many people did. For people in the audience, Don's editorial actually reached a download of 23,000 downloads in a record speed. So that's a very, very amazing editorial. I recommend everybody in the audience to read it. So with that, Don, what's actually your favorite molecule? As a chemist, we're always kind of attached to molecules. So I wonder, what's your favorite molecule and why would you say it?
I mean, that's a great question. And I mean, being a chemist, there's lots of molecules, because let's face it, molecules are artistic creations. There are little sculptures that we make. But believe it or not, my favorite molecule is going to be so disappointing for you. It's
Let's hear it.
valproic acid.
Oh, okay.
And you go, valproic acid, it by no means is it a product of rational drug design. It was discovered, completely by accident. But what I like about it is it certainly lives up to the engineering design principle of keep it simple, stupid. It shows the power of an exceptionally simple molecule. And because valproic acid is such a simple molecule, and yet it has such profound pharmacologic effects. And back when I was seeing a lot of patients also had epilepsy, so many of them responded so beautifully to such a simple agent. It did so well. The fact that it is simple and easily produced, I think, addresses what I regard as an important issue in med chem, and that is the diseases that we deal with are global. And I think that global diseases need global solutions. And when I see really expensive biologics and things coming out, that's nice. And yes, it's important, and I agree. But 80% of the world doesn't get access to them. And so I think that having small molecules, having simple small molecules that are easily made, certainly gives us the opportunity for better global penetration. And the final thing that I like about valproate is it's one molecule with multiple receptors. And I think that that's an opportunity that we don't exploit enough in small molecule med chem, especially neurologic ones, because our molecules are even simpler because we have to get across the blood-brain barrier. And, so if we can take a molecule, and unlike a biologic, which is a sophisticated molecule that has a very specific receptor that it interacts with, if we can build in and take advantage of the simplicity of our molecules by having them interact with multiple receptors. And I know there's SAR issues. I know there's dose response issues. There's challenges, but they still can be addressed. And, if we can get one molecule that hits multiple receptors, that's powerful. That's exploiting the benefit of our approach.
Nice. That's, again, I think a surprising answer, but that's an answer I think you could only give with your perspective as a clinician and a chemist. So love that. What are you excited most about in drug discovery today? Is there like a technology or anything that you're like really excited about? I mean, I know we had talked a little bit about how neurologists, can't do much with the tools that they have today. So I wonder, what technology are you really excited about today? Or
I don't know if it's a technology. I'll say what I'm excited about is what I think of as renewed hope in the area of Alzheimer's. It has been a no man's land of failure for so long. And with lecanemab and this week with donanemab coming out, they're not the cure, but they're steps, small steps with modest, efficacy, but they're steps in the right direction. And I think that if pharmaceutical partners can see this and go, maybe this is an area that we should be back in and we're interested again. If this kindles interest and fires up the interest in neurodegenerative disease and promotes continued research in it, and I think that's what it's going to do, I think that's exciting. So finally having something that may offer some even small measure of success in Alzheimer's is a huge step forward. And I hope that this is just foreshadowing a very, very promising future.
awesome. Yeah, no, that's very, very exciting. Yeah, absolutely. So, Don, you had a really long-standing career, so and you were very successful in everything you did.
Or still am, hopefully. I'm not that old.
So I wonder if you could go back to the beginning of your career, imagine it's 1981 or something like that, and
Yes.
then
Yes.
you
When I had hair. Yep.
what advice would you give yourself during that time if you could start all over again with your career?
Yeah, I get asked that question a lot. And people say, you've spent a career doing a day, day and a half clinical a week, and 3 to 4 days chemistry a week. If you could do it over, would you just go back and do all chemistry? And no. I mean, love chemistry, and it is, the power that makes drugs. But I've always thought that bringing the clinical element to this has been important, and has given me a perspective that I've found, particularly useful. So from my point of view, I've always would have combined the two. Students always ask me, well, what advice can you give me? And my advice is learn as much organic chemistry as you can and learn some more. You can't learn too much organic chemistry, because ultimately, as I said, we're sculptors who make molecules, and having the tools to make those molecules, to give them the beauty that they need, is essential. And becoming the best damn organic chemist that you can be, I think, is crucial. In my own point of view, if I go back, I think that I would like to have come to grips with the notion that there's not going to be a single magic bullet for Alzheimer's. I wish I would have come to grips with that concept earlier in my career. This is too complicated a disease. We're not going to say, here is the pill, that is going to do it. I think we're going to recognize more subtypes of Alzheimer's and the need for, polypharmacy, multiple meds, and individualizing into patients. And I think that would have changed my thinking and framed my approach differently had I come to grips with that concept earlier.
Yeah, but I mean, you did come to grips with that concept, and I think you were, one of the first people still who came to grips with that concept. And
Yes.
I think I can very much relate to what you said about the organic chemistry. As a trained organic chemist myself, of course, you can never learn enough organic chemistry. Such a big field, it's worth knowing all these reactions to make all these molecules 100% done. Thank you for
Yep.
your advice. Before we get started, one last question I wanted to ask you, Don. What do you like most about Drug Hunter?
All right. Um, two things. And I suppose they both begin with P. One is an emphasis on people, and secondly is an emphasis on practicality. So first is an emphasis on people, and I mean there was and this was triggered by a LinkedIn posting that came out from you last week. Which you emphasize, yeah, we make molecules, but it's people who make molecules. And science is a very human activity. The greatest strength of science is that it's human. Probably the greatest weakness of science is that it's human. But it's people and their passions who drive it. And I think understanding people, understanding their motivations, is a key element to understanding medicinal chemistry. And the people component is also crucial, that we're making drugs for people. We're not making drugs for a biochemical assay. We're making drugs for people. And I think that's important. And the other P thing was practicality. I mean, the advice that comes out from you guys is practical. It's useful. I was following some recent conversations, on log Ps and things like that. And you just go, this is just pragmatic, brutally practical, useful advice. That it's nice to have a place where this comes from, a trusted source of information. And so, yeah, Drug Hunter is certainly valuable.
Awesome. Thank you so much, Don. Really appreciate it. And yeah, thanks for reading my LinkedIn post too on this. I'm very passionate about this too. I'm very much a people person, as hopefully you and others can tell. So yeah, before we get started with Don's presentation, I wanted to get very briefly share a highlight that I learned in our April patent highlight. So this is just a tip that you can use maybe to improve your projects. So if you're working in the CNS disease area, in our April patent highlights, there was a brain penetrant TYK2 inhibitor that was patented by BMS. And it differentiates here in its head group. So this is a 1,2,3 triazole, and our earlier compound here from alumni has a 1,2,4 triazole. And while they're structurally very, very similar, what they're differentiated by is their brain to plasma ratio. So as you can see here, the 1,2,3 triazoles have a brain to plasma ratio of 3, whereas the 1,2,4 triazoles only have a brain to plasma ratio of 0.5 to 1 to 0.15. So this is maybe a trick that you can use in your next project if you needed to improve the brain penetration. And TYK2 is also a target in neurodegenerative diseases such as Alzheimer's. And I guess that is the perfect segue to Don's presentation. So Don will talk to us about minds and molecules today, lessons learned from Alzheimer's diseases. And we're very excited to hear your perspective on the pathology of Alzheimer's disease, of current treatments, and new innovative therapeutic approaches. And yeah, thanks again, Don, for being our guest today. And I'll leave the stage to you now.
Well, thank you. And thank you for the opportunity to present today. What I'm going to talk today is about how I think about Alzheimer's disease. You know, I tend to think of Alzheimer's disease as an immunologic disease of the brain. And I'm going to, you know, share some of my thoughts on this approach. I'm not saying I'm right. I'm not saying I'm wrong. My goal here is, you know, for goodness sakes, just let's think about what we're doing. We've been hammering away at Alzheimer's for decades with not a lot of success. So, you know, when we start thinking about this disease, when we start approaching this disease, you know, let's keep our minds open. Let's think about the approaches that we're using. Let's think about how we understand this disease. First of all, I'll just start off with my disclosure. I'm involved with our most recent startup company, a company called Treventus, which is focused on primarily tau, and we've partnered with Takeda. But today I'm going to be really focusing on some work that comes out of the academic portion of my lab, much more on the immunological aspects. And this is an aspect of our work that is not partnered with a pharmaceutical partner. So Alzheimer's disease. I mean, traditionally, we regard this as a problem of memory and cognition. And the problem with memory and cognition arises from widespread brain atrophy, in which the typical brain drops, you know, almost half a kilogram of brain. And yes, this understands it at a gross neuropathological level, but it certainly doesn't help us in devising therapeutic approaches. In terms of small molecules, you know, we all appreciate that attempts to come up with disease-modifying small molecules has not been successful to date, and that the small molecules that we do have out there, such as those shown on this slide, really are symptomatic agents only, primarily targeting, you know, acetylcholinesterase, but they really aren't affecting the natural course of the disease. And this is an area where we definitely need to improve. Some other aspects of the problem of Alzheimer's that, you know, I really think needs emphasizing is that first, it's a lethal disease. It's a terminal disease. If you get Alzheimer's, you're going to die from Alzheimer's unless you die from something else first. Secondly, this is a disease whose numbers are expanding tremendously. Currently, there are 52 million people worldwide living with Alzheimer's, but by 2050, we expect this number to exceed 150 million. So this is rapidly evolving into a huge market, and it's a market for which we have no cure currently. We've got nothing, you know, that really is truly a curative agent. You know, why is that? And it's difficult to really know. If we look at how conventionally we've been thinking about Alzheimer's over the recent decades, we've always regarded it primarily as a protein misfolding, and we've talked about the amyloid hypothesis, where there is two peptides, amyloid and tau, primarily amyloids, the ones that receive the majority of the attention. And, we regard it as a pathological protein. It's not supposed to be there. And when it is there, that's abnormal. It aggregates. The aggregates are neurotoxic. They destroy neurons, and that then leads to, progression of the disease. This has been, the prevalent view, and conceptually and intellectually, it's not a bad hypothesis. The only problem with it is that it really hasn't led up to major curative agents, major steps forward from a therapeutic point of view. As is well appreciated over the past 30 years, the majority of small drug trials have failed. All of them at disease modifying have failed. And yes, we do have recently lecanemab and donanemab, the biologics, which do target amyloid and which have shown some very modest improvements in the face of, significant side effect profile. And so it certainly suggests that amyloid is part of the story, but it's not the entire story. And it gets back to this issue that I was mentioning. There may not be a single magic bullet. We may be looking at a magic shotgun blast here as we try to address Alzheimer's disease. So I think that we really need to go back to first principles and, rethink this disease, rethink our approach to it, rethink how we, try to understand it and try to use this understanding to develop a therapeutic approach. And this is a bewildering exercise. If we look, you can find very passionate arguments, saying it's an immunopathy, it's a mitochondriopathy, it's due to membranes, it's due to synaptic toxicity, there's a metal dyshomeostasis. There's lots and lots of different, well-argued and, probably viable approaches to coming up with novel therapies for treating Alzheimer's disease. The brain's complicated, Alzheimer's is complicated, the number of biochemical pathways involved and implicated is huge. And so, we really are confronted with a bewildering array of different receptors that we could be going after. One of the other, essential elements of this is amyloid itself. There are people who say the amyloid hypothesis is just dead wrong. We should reject it. And we should just move on in a post-amyloid sort of era. And there's others who go, no, that's not right. Amyloid has a role, and I think that's justified in part by lecanemab and donanemab. And but maybe we should be thinking and rethinking the role of amyloid and the role that it plays in Alzheimer's. And that's what we've been doing. One of our approaches is just sitting down and rethinking about beta amyloid. And what we did is we started off and just did some molecular dynamic simulations. Let's take beta amyloid, put it on top of a model membrane, and see what it does. And so, when we did that, what one rapidly finds is that amyloid initially seats itself on the top of the membrane, and then a portion of the amyloid peptide wiggles its way through the membrane, ultimately breaching the membrane. And it can do this singly, or it can certainly do this much more in a neurotoxic fashion if it's in an oligomeric fashion with a number of amyloids linked together. And so, if we look at this, what one sees is that the N-terminal portion of beta amyloid comes down, seats itself on the surface of the neuronal membrane, certainly with this HHQK domain, cationic domain, establishing electrostatic interactions with the membrane surface. And then the C-terminal portion, which is much more lipophilic, actually folding itself in through the membrane and enabling this breach to occur. And so, that is something which certainly emerged from our very initial simple modeling. But that was with a fairly homogeneous, straightforward, simple model of a neuronal membrane. Neuronal membranes aren't simple. They have a fair degree of heterogeneity and complexity within their structure. So first of all, if we start to include other types of lipids, not just the standard sorts of lipids that were used in our initial modeling. We start bringing in phosphatidylserine, phosphatidylinositol phosphates, cholesterol, etc. Then you find that certainly introducing, as I said, these particular lipids makes them more susceptible to this membrane penetration effect that I described with the C-terminal portion of amyloid perforating through the membrane. And interestingly, these sorts of lipids are much more densely located within the synaptic region of a neuron, because certainly the overall lipid composition of a neuron is not homogeneous over the whole neuron. There's a lot of heterogeneity over different regions, and the synaptic region is much more susceptible to this penetration by A beta. And this is actually in keeping with what the biologists tell us, that synaptic regions seem to be more susceptible to A beta induced toxicity. Also, if we start getting away from just lipids and look at other molecules, and so gangliosides, for example, GM1 decorates the outside of the neuronal membrane and do a number of glycosaminoglycans, such as heparan sulfate or chondroitin sulfate. And here again, A beta establishes very tight interactions and complexes with those particular molecules, as I said, which decorate the external surface of a neuronal membrane. So if you look at this, you go, A beta penetrates neuronal membranes, piercing them preferentially at a synapse, but it also will establish favorable geometric and energetically favorable interactions with GM1 and with glycosaminoglycans. And so, there are very distinct interactions between A beta and the neuronal membrane in these molecular modeling exercises. So from this comes the realization that A beta exhibits antimicrobial activity. Perforating a membrane is how antimicrobial peptides work, and certainly viruses frequently attach to cells by binding to GM1 or binding to glycosaminoglycans. And so by doing this, A beta has an antimicrobial effect, certainly both antibacterial and antiviral. And by no means are we the first people to have made this realization. The understanding and appreciation that A beta is an antimicrobial peptide, is a notion that's been out there for a number of years. And we, like others, went ahead and actually did the experiments and certainly demonstrated that A beta has very significant antibacterial and antiviral properties, when we actually do the experiments in the laboratory. The other interesting thing is that if we played with concentrations of copper and zinc, we could increase the antimicrobial activity. And if we also played with concentrations of cholesterol, we could also interfere and change the antimicrobial activities. And this is in keeping with normal antimicrobial peptides. It's appreciated that antimicrobial peptides certainly become much more toxic when they're complex with copper and zinc, etc. So this is fitting into a nice story, of an antimicrobial effect for A beta. So then this is part of this rethinking about A beta. Maybe we shouldn't be thinking about this as a pathological peptide. It's a physiological peptide. It's there for a reason. It's antimicrobial, and if it's an antimicrobial, then it's an immunopeptide. It's contributing to the immune system's response to threaten insult or infection. And so, let's start rethinking about amyloid, not as a pathological thing, but as a physiological immunopeptide that plays a role within our brain. And if it is an immunopeptide, what type is it? And most people go immediately, it's an antimicrobial peptide. That's what it does. And antimicrobial peptides are part of the innate immune system. Rather, we have looked at this and have questioned this and pushed it a little further. And, based upon a number of molecular modeling exercises and comparing it with known cytokines, etc., we began to wonder, is it more than just an antimicrobial peptide? Does it reach the threshold for actually being a cytokine? We looked at a large number of cytokines and described them by a number of descriptors, and in fact, determined 30 properties which determine if a peptide is a cytokine. And then we demonstrated that beta amyloid indeed satisfies all of these properties. So from my point of view, we really could think of beta amyloid as being a cytokine and as being a cytokine with antimicrobial activities. And there is a subclass of cytokines referred to as kinosidins. Kinosidins are cytokines which are significantly antimicrobial. And so I am suggesting, rightly or wrongly, that beta amyloid is a kinosidin. It's a physiologically released immunopeptide that for some reason becomes pathological. And so what are the implications of that? So, if we're thinking that beta amyloid, is a physiological peptide that goes rogue, that something goes wrong and it suddenly becomes toxic to neurons, that is how we're starting to reshape our thinking about beta amyloid and its role in this disease. So what we did is we went back and began to look at this again. And, if you do the molecular modeling of beta amyloid interacting with neurons, and you do the molecular modeling of beta amyloid interacting with bacteria, it looks very similar. They're behaving in similar fashions. So here is an antimicrobial peptide that perforates the membranes of bacteria, killing them, that analogously perforates the membrane of neurons and kills them. But it's killing neurons preferentially, because if we are spilling A beta in the presence of other glial cells, microglial cells or astrocytes, it's not killing them, it's activating them. And so, you sit and you go, does A beta mistake a neuron for a bacteria? Is this why it has the opportunity to go rogue? Because it can't tell the difference between a neuron and bacteria? And I mean, you look and you go, both neurons and bacteria have transmembrane voltage gradients, and other things that make them susceptible. And certainly, we started to produce some electrotopological maps in which we mapped out the charges and mapped out the lipophilic areas, et cetera. And if you really, look at a neuron, particularly in the synaptic area, and you look at a bacteria, there is surprisingly degree of overlap here. And so what I'm suggesting is that beta amyloid, when it's out on its search and destroy mission looking for bacteria and it's flying over a target, it can't differentiate a neuron from a bacterium. And that is the opportunity for a physiological peptide to go rogue, to go bad, to go pathological. That's nice. But what it doesn't explain is why this is a chronic disorder. That's acute. I mean, that's the innate immune system that's rapidly dealing with an infection or something. But Alzheimer's disease is a disease that takes decades to unfold and to progress. And what I've just described is this confusion in not being able to differentiate a bacterium from a neuron. That's not a chronic process. That's an acute process. However, A beta, as I mentioned, binds to gangliosides such as GM1, forming a fairly tight complex. And when the cell becomes necrotic and broken apart, it actually releases these A beta GM1 complexes, which diffuse to nearby adjacent neurons. And that triggers these neurons to start producing more A beta. And so we actually get in a chronic cycle that A beta is released, it kills a neuron and binds to the neuron. The products released from this necrotic neuron go to adjacent ones, trigger them to produce A beta, and we got a cycle going. And so we actually verified this experimentally. We took neurons and killed them by necrosis, killed them by apoptosis, and demonstrated that the necrotic products that come from a necrotically killed neuron are able to go to adjacent neurons and able to trigger them to produce A beta to sustain this self-sustaining chronic cycle. So does this immunopathic mechanism make sense? Are we allowed then to call this an autoimmune disease? And this gets me in so much problems. And here I have immunologists going, no, 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 you're wrong. This doesn't make sense. And I have other people saying that it does. And I use the word autoimmune here with trepidation and with caution. And please note that I put it in quotation marks. Classically and traditionally, as we understand autoimmune diseases, it is a disorder of adaptive immunity. It's a disorder in which we have antibodies which mistakenly recognize host antigens, and so we have antibodies targeting us. I'm not invoking antibodies here at all. I'm invoking that we have A beta as a cytokine that specifically recognizes neurons. Normally, when we think of innate immunity as going wrong, we think of it in terms of an autoinflammatory response, such as that cytokine storm, et cetera, where there's indiscriminate killing of cellular elements because they're nearby. This is not indiscriminate killing. This is a focus-specific killing of neurons because neurons have electrotopological similarities to bacteria. This is a focused attack by the antimicrobial kinocyte cytokine A beta upon neurons. So I use the word autoimmune, but it's an autoimmune disorder of innate immunity rather than adaptive immunity. And, as I said, classically in immunology, this is an incorrect use of the word. But I apologize for the words on this, but this basically summarizes what I've been saying. In response to various stimuli, infection, trauma, ischemia, pollution, depression, whatever, A beta is released as a cytokine that triggers an innate immune cascade in which A beta exhibits both immunomodulatory and antimicrobial properties. Whether bacteria are present or not, it's still going to demonstrate antimicrobial properties. And this results in a misdirected attack upon self neurons arising from analogous electronegative surface topologies between neurons and bacteria, rendering them similarly susceptible to membrane penetration by A beta. The resulting necrotic breakdown products diffuse to adjacent neurons, eliciting further release of A beta, causing this to be a chronic, self-perpetuating cycle. And therefore, I'm suggesting that AD emerges as a brain-centric autoimmune, in quotation marks, disorder of innate immunity. Key points here. Beta amyloid's a physiological immunopeptide, a kinocytin. It's physiological. Beta amyloid can't differentiate between neurons and a bacterium when released, and therefore Alzheimer's possibly should be considered as an autoimmune disease of innate immunity. The other key point that I will mention here is that please note that this way of thinking about Alzheimer's disease has come from us as chemists, as medicinal chemists. And I think in the drug discovery process, we often think of this in a linear sort of fashion where biologists, put forward an understanding of the disease, identify, reasonable receptors, and then the medicinal chemists come in and target that receptor and come up with molecules that are able to have the appropriate pharmacodynamic properties to interact with that receptor. But we as chemists are people who think about things at a molecular level, and having molecular level thinking, has a useful feedback to the biologists in understanding how they regard the disease. And we can make contributions in the molecular understanding of the disease that extends beyond, inventing drugs. It also uses us to contribute to understanding the biochemistry of the diseases that we are endeavoring to treat. So in the last few minutes, what are the therapeutic implications of this? The immune system is one of the most regulated systems, physiologic systems that we have in our body, and rightly so. It is filled with checks and balances because we want it to be just in the right balance as it helps us in our daily struggle against microbiological toxins that are, constantly in our world, and other insults to the structural integrity of us as a host. And so you go, if this is primarily immune disease, are there endogenous regulators of innate immunity within the brain that we could be exploiting here for a therapeutic opportunity? And so Also, this was triggered a bit by the notion, say, of steroids. If somebody comes in with a conventional autoimmune disease, a disease of the adaptive immune system, and they're in a rough way in the emergency department, we give them large doses of steroids. Steroids are an endogenous regulator of adaptive immunity. Can we come up with an endogenous regulator of innate immunity, which would let us try to address what's going awry here in Alzheimer's disease? So we did initially an in silico screen of more than 1,100 small molecules endogenous to human brain. And interestingly, we came up with 3 amino acids, taurine, L-tryptophan, and L-arginine, and metabolites thereof as agents which seem to have a role in regulating the innate immune system. We repeated this screen this time in vitro, and actually did the same more than 1,100 molecules in an in vitro screen, seeing if we could identify I'll call it an endogenous anti-A beta or anti-Alzheimer's molecule. Once again, this identified primarily tryptophan and arginine. And, we thought how clever of us, but not really. If you look at the oncology literature, and certainly they are experts in trying to manipulate the immune system for a beneficial therapeutic outcome, they realized years ago that tryptophan and arginine are valuable targets in trying to modify the immune system in a therapeutic way. And so here we can learn from the world of oncology and take some of those lessons and bring them over to our efforts in Alzheimer's disease. And so, we went, yeah, just as steroids have a role as endogenous regulators of adaptive immunity, the clearly evolutionarily much older innate immunity should have its own endogenous regulators. And not surprisingly, amino acids emerge here. If you go through the evolutionary biology, it would make sense that some of the earliest building block molecules might also serve multiple functional roles as regulators of the primitive innate immune system. And so we decided to look at all 3 of those. Initially, way back when, we actually targeted taurine and made a large number of analogs of taurine, took one of these to a phase 3 trial as the compound trimiprazate. It failed, and we personally abandoned taurine and didn't take it any further, or the homotaurine story. There is another drug company out there now, Alzion. I'm not involved with them, but they have picked up the torch and have taken it and have continued very admirably, to continue to work in this area and to make important advances in this area, and are doing some very interesting work at improving homotaurine and turning it into a viable therapeutic entity. We, on the other hand, have looked at the other amino acids and have been focusing on tryptophan and arginine. And I'm just going to say a few words about our efforts in the area of tryptophan. So what we have looked at is, as I mentioned, a number of tryptophan metabolites had a degree of efficacy when we did our initial screening. And so we have been producing analogs of these metabolites. And we've also looked at inhibitors of IDO. IDO is the enzyme, which is the first step in the catabolic breakdown in one of the catabolic pathways for tryptophan degradation. And certainly in the world of med chem, targeting an enzyme is always an attractive target. It's a comfort area for us. It's an area where certainly there's been a rich past history of coming up with enzyme inhibitors. So initially, we produced a lot of compounds as analogs of tryptophan metabolites, and have had compounds here which certainly had interesting in vivo and in vitro and in vivo activity in a number of different mouse models. We've always had some PK problems with this, but this is an area of interest ongoing for us. And as mentioned, the other area of interest for us is IDO and targeting IDO with enzyme inhibitors. And this is an active area that we're also working on. as we trying to come up with agents which might be a brain penetrant IDO inhibitor. Once again, lots of lessons from the world of oncology. There are lots of agents which have been developed as IDO inhibitors for cancer indications. But if you look at these, they don't cross the blood-brain barrier. So what we have been looking at is trying to come up with brain penetrant IDO inhibitors. And once again, we've produced a fair number of compounds. We've taken some of these through in vitro and in vivo, and certainly have demonstrated that one of our IDO inhibitors, certainly in some of the standard transgenic mouse models, has improved memory and learning, and have also been associated with ameliorating some of the anxiety and behavioral disorders associated with Alzheimer's in these particular mouse models. So in summary, I'm putting forth an idea, and if nothing else, I hope it just triggers you to think about this disease. We are suggesting that this is a disorder of innate immunity, and that it is subject to potential endogenous regulation by tryptophan and arginine, and this could represent certainly a new way of trying to come up with small molecule therapeutics for this disease. The other thing I'm just going to say in closing is, if we think that A beta is a cytokine, does this enable us to rethink about drugs like lecanemab and donanemab? Are these antibodies that target a cytokine, which would make them not dissimilar from a lot of other antibodies out there, like Remicade, which certainly targets TNF alpha. And is this a way of potentially thinking of the therapeutic benefits that we are getting out of lecanemab and donanemab? I'll close by bringing this full circle to the questions that Dennis asked at the beginning. And I said, people are important. And when we do this, we have to remember that there are faces in front of the molecules, there are faces in front of the beta amyloid, the tau, the neuroinflammation in the brain, and it is these individuals for whom we are doing this work. As always, making compounds, as everyone on this call knows, takes a lot of time, a lot of effort, and I describe myself as merely the eye candy for our lab. Sorry. And it takes a large number of people to do this work, and I acknowledge them. It also takes a number of support, and I acknowledge the many various institutions which have supported our work over the years. To them, I am very grateful. But once again, please remember why we're doing this. We're ultimately doing this for our patients. Thank you ever so much.
Thank you so much, Don. That was an amazing presentation. Yeah, it's like so surprising to me. I mean, I hadn't thought about beta amyloid in that sense, that it, I mean, I'm not an expert in Alzheimer's by no means, but that it can act as a cytokine is certainly something that was kind of really unexpected to me. So yeah, we do have a few questions from the audience already. So first question is here
only
from...
nice questions. Only nice questions.
Only nice. Yeah. Only nice questions. The first question is, if you could elaborate a little bit about the conditions under which beta amyloid is not able to differentiate neurons and from bacteria. I think you talked about it a little bit, but is it like any condition or?
So yeah. What I'm saying is that antimicrobial peptides, they have activity against both gram-positive and gram-negative bacteria, a bit better against gram-negative bacteria because you're not having to deal with the cell wall. But I mean, if you look, they come down and bacteria talk to other bacteria. There's, we recognize there's bacterial films, and we recognize that bacteria don't live in isolation. When you have a lot of bacteria, they are functioning as a cooperative group, with bacteria talking to other bacteria as they plan things. They act like a little brain. A film of bacteria act like a little
Interesting.
brain. They have ion channels. They are sending messages to each other. Hell, they even use acetylcholine as a messenger.
Really?
Um, and so, you take that and you go, that's how our brain works. So there's lots of different types of neurons, and no, we haven't looked at all the different types of neurons, but we appreciate, that neurons are susceptible, and the gross pathology that pathology teaches about Alzheimer's disease certainly suggests that all brain regions aren't equally susceptible to A beta. And no, we haven't done that to prove that correlation, but certainly our modeling does demonstrate that synapses are more susceptible than the other areas of the neuronal membrane. And so it supports that. But no, fair question. But this is early times for this hypothesis for us. It needs to be elaborated more fully. But as I pointed out, it's not like an indiscriminate killer. Because when we have A beta out there and there's neurons and microglia, the microglia cells are activated. The neurons are killed. And so that's why I'm making this point that neurons seem to be specifically susceptible.
Got it. Got it. Yeah, maybe I have actually a question myself about the targets. So you talked about IDO enzyme inhibitors, right?
Yep.
So I wonder how are you thinking about maybe different targets in any catabolism of amino acids, or what do you think is how could you identify other targets for Alzheimer's disease? It's just a question that was on my mind the entire time.
Yeah, because tryptophan is such a miserable amino acid. If you go through the catabolic breakdown products of tryptophan, which is one that we're mainly focused on, some metabolites are neurotoxic. They're bloody awful. And some metabolites are beneficial. And so, if you work your way through the path and you go, which enzyme should I be hitting? You got to be careful, because you don't want to start, suddenly cranking up levels of quinolinic acid, or something that's going to be horrendously neurotoxic. And so, that's why we hit IDO. It's up at the top. And therefore, we're shuttling everything more to the serotonergic pathways, which has its benefit. There's this fascinating overlap between depression and dementia, depression and Alzheimer's disease. Are they one and the same disease? Is geriatric depression and Alzheimer's, are they connected? I mean, certainly depression is a risk factor for Alzheimer's disease. If you talk to a geriatric psychiatrist, they'll tell you that patients who never had depression in their life, who onset with depression in their 60s or 70s, they got dementia. This is just the first manifestation of it. And so, putting this all together, it's still part of the story. It still makes sense. Yep.
Yeah, no, very interesting. Thank you. Yeah, a few more audience questions. How does the autoimmune beta amyloid hypothesis and mode of action you describe tie in with the tauopathy associated with Alzheimer's disease? And has any research gone into understanding whether tau aggregates can induce this beta amyloid autoimmune reaction?
Okay, excellent question. And this is where I go, next question. No, that's I mean, clearly this is an area about which we're thinking a lot. And that question is a question that we pose to ourselves. Do I have a good answer? No. I mean, certainly we appreciate that over the course, A beta is busy misfolding for decades before tau somehow gets triggered. How this fits into this is not clear. I will point out that tau is also antimicrobial. And so, so tau is antimicrobial. Tau definitely, although we don't talk about it, also has immune properties. And so that's how we're trying to weave the stories together. We haven't done it successfully yet. It's a work in progress for us. But yeah, no, excellent question. And as I said, it's a work in progress as we try to sort through that mess.
Awesome. Yeah, and research, sometimes we don't have all the answers, but that's why
No,
it's called
especially in
research, right?
Alzheimer's. Yeah, yeah.
Awesome. Another question here from Geoff Tramner in the audience is the idea of amyloids as cytokines is an interesting idea. Could you comment on the potential of amyloids being an intentional cytokine versus a mistake in cytokine production?
Okay, so, and you use the word amyloids, which I appreciate, because beta amyloid is only one amyloid. There's lots of different amyloids. There's systemic amyloidosis. There's amylin in the pancreas, which of course is implicated in type 2 diabetes. You know, we have looked at trying to understand that there's this relationship, of course, between type 2 diabetes and Alzheimer's disease, you know, with some endocrinologists saying that we should really think of Alzheimer's disease as type 3 diabetes. I personally would like to think of diabetes as simply Alzheimer's of the pancreas. So in my dreams, I think of amyloids as perhaps unappreciated immunopeptides, and that this extends beyond beta amyloid. This extends to other amyloids and, you know, to amylin, the culprit in type 2 diabetes, and that, you know, they are a vibrant but underappreciated part of our immune system, which just because of the unique electrical properties of neurons become, you know, susceptible in Alzheimer's disease. You know, and if you look at this from an evolutionary point of view, having a potent antimicrobial peptide in your brain is useful when you're young. It protects you from a whole lot of things. And let's face it, if it's going to take 30, 40 years to kill you when you're old, from an evolutionary point of view, you've already done your reproduction. You know, from the cruelty of evolution, you're now expendable. And so, you know, it's a small price to pay to have your brain protected in youth.
Got it. Got it. We have many more questions. Maybe this one here. Is there such a thing as a cerebral microbiome, given the recent findings and links between immunology and the microbiome that from Rudy? Could there be a link between the microbiome environment and the expression of antibacterial cytokines in the brain?
Yes, yes, yes. There are some I mean, I have not done the work by any means, but yes, there are papers coming out suggesting that there is a cerebral microbiome. We've always, you know, fancied the brain as being a very sterile place, but, you know, it's not. And so probably we do have ongoing microbial occupants of our brain. So yes, there would appear to be a cerebral microbiome. And if we go to the microbiome in general, particularly from a gastrointestinal point of view, where, you know, it has its full expression, novel therapeutic approaches to Alzheimer's are exploiting the gut's microbiome. There's reason to believe that tryptophan metabolism in the gut is influenced by the microbiome, that metabolites coming out of our gut's microbiome have a direct effect on brain, and that, yes, this could also be an important contributor to the progression of Alzheimer's disease. Yeah, really neat question. And I think this is a really neat area.
Awesome. Maybe I think we're on the top of the hour. One more question to wrap it up. I see one interesting question here in the chat. Do you think the NRLP3 inhibitor or CGAS STING inhibitor work for preventing Alzheimer's disease, since it could be acting on an anti-inflammatory signaling?
Yes. So once again, good question. There are so many different approaches to neuroinflammation. So, you know, as I said, I think it's going to be a magic shotgun blast, not a magic bullet. An anti-neuroinflammation agent, I think, is going to be an important contributor to the ultimate successful cocktail. There are so many different viable receptors in the area of neuroinflammation. And the inflammasome mentioned is certainly one of them. There's a lot. I mean, TREM2, of course, is attracting a lot of activity. There's a number of them. And I think that regulating the immune system in the brain, not turning it off, I'd rather think of this as a thermostat. We just want to turn it down. We don't want to turn it off. And that there are a number of targets that'll let us do this. And certainly the inflammasomes are one of them. So yeah, I think that is a potentially worthwhile target.
Awesome. Thank you so much for your perspective. That was such an enlightening talk for me and the audience. We have many more questions, unfortunately, that we can't get through because we're already on the top of the hour. So, Don, thank you for an incredibly
Well, thank you.
inspiring
That was fun.
presentation. Yeah, super fun. And yeah, thanks to the audience for the questions and the engagement. We really appreciate it. And we will see you at the next event.
Thank you ever so much.